Hello. On behalf of the Biochemical Society and Portland Press, I'm really pleased to welcome you to today's webinar, which is part of our Biochemistry Focus webinar series. As you most likely already know, topics in this series include different research areas in the molecular biosciences, as well as practical sessions to support career development. Each webinar indeed gives you the opportunity to ask questions via text, and we also welcome suggestions for future topics and speakers to feature in our webinar series. And for this, please see the Biochemical Society website for more details. To start with, I will introduce myself. I am Luminita Paraoan. I'm Professor of Molecular Cell Biology, leading the group of Ocular Molecular Biology and Mechanism of Disease at Edge Hill University. I will be chairing this webinar, in which we are celebrating the 60th anniversary of the Colworth Medal, kindly sponsored by Unilever. The Colworth Medal, as you know, is a prestigious annual award presented for outstanding research by an early career biochemist of any nationality who has carried out the majority of their work either in the United Kingdom or the Republic of Ireland. In this series, we bring together some of our past winners from across the decades to discuss their careers, achievements, and give us their advice. Today, I am truly honored and delighted to be joined by Emeritus Professor Laskey, CBE, Professor David Lilley, Professor Akhilesh Reddy, and Dr. Sarah Teichmann. Before I will ask each one of them to introduce themselves, I would like to remind everyone that questions are encouraged and indeed possible to be received throughout the webinar and will be asked at the end of the sessions and the discussion. Please type your questions into the box as shown in the image on the screen, and we will try to answer as many as possible as time will allow us. Now, so let's start. I will ask each of our panelists to introduce themselves, and although I'm sure their areas of research are already well known by the majority of attendees, please include that in your brief introduction. We'll start with, in the order of the award, therefore, Professor Ron Laskey, the Colworth Medal awardee in 1979. Please, Ron.
Thanks very much. I'm almost ashamed to admit that I didn't start life as a biochemist. I started as a zoologist and did my doctorate on transplanting nuclei from adult cells into eggs from whose nucleus we'd removed. And I then went on to study various aspects of DNA viruses, of animal viruses, at what was then Imperial Cancer Research Fund and shortly to become Cancer Research UK in those days. And from there moved to the Laboratory of Molecular Biology, MRC Laboratory of Molecular Biology, where I spent the next 10 years. And it was during that phase that I was awarded the Colworth Medal, which to my shame, when the letter arrived, I was walking past my mailbox, picked it up and put it in a back pocket without opening it, and forgot about it for an embarrassing length of time before discovering that it contained a rather nice bit of information about the Colworth Medal. But the award there was for two things. One was work we'd done on chromatin assembly in vitro, as a byproduct of which was the term molecular chaperone, which we introduced to describe how it functioned. But the other thing was radioisotope detection, which was an aside. It only took 2 or 3 months of research time, but finished up producing my 2 most highly cited papers, which remained my most highly cited papers for the rest of my career, on how to detect tritium in acrylamide gels by fluorography, and how to improve detection of P32 and gamma emitters by X-ray intensifying screens at -70. But from there, I moved to the Department of Zoology in Cambridge, and then showed an appalling inability at career progression, because I stayed there in the same post for 29 years, though with the digression of helping construct 2 new institutes. And during that time, we worked on control of DNA synthesis in animal cells and human cells in particular, and on how proteins entered the nucleus selectively, and finished up looking at how mRNA selectively exits the cell nucleus as well. So it's been a rather wide, divergent history of things to do, but fun all the way, and probably becoming more biochemical as I progressed from my zoological start in Korea.
Thank you, Ryan. Can I ask David for, just to introduce yourself briefly and the main area of your research?
Right. So well, I'm David Lilley. I think I've moved further than Ron actually, because my PhD is in quantum mechanics. But that's a tradition, I mean, the origins of molecular biology. I guess most people either came from physics or phage genetics, and so I'm sort of half of that tradition. But anyway, I forget now. I must have won the Colworth Medal about 40 years ago, I think. So a lot of water's passed under the bridge since then. But I'm a biophysical chemist, and actually I guess I won the Colworth Medal for the work I did on the topology of DNA supercoiling, that sort of thing. All my career, really, I've spent in nucleic acids. So I'm a biophysical chemist studying nucleic acids from a structural, biophysical, and above all mechanistic point of view. I mean, one of my strong interests, for example, is in ribozymes, which are enzymes that happen to be made of RNA, not protein, and particularly the mechanism. I want to get under the chemical hood of that and understand how they work. So I've spent really the last 43 years, I think, in Dundee. That's probably not a brilliant career move to stay in the same place. And Ron said he spent, what, 29 years in the same place? 43 here. If you want to get a good salary, move every 10 years. Don't do what I did. But I've just loved being here, and I never saw any great reason to move. But the other thing is, I actually spend quite a lot of time in China these days. So I have a lot of connections with China. I used to say at one time I probably gave a fair fraction of the seminars I gave in French. These days, some fraction of the talks I give are in Chinese as well. So, but I have wonderful collaborations. Well, we can come on to this later, I'm sure, but in China. But so, I'm sort of somewhere but delocalized between here in Dundee and places like Xiamen and above all Guangzhou and Beijing in China.
Thank you, David. Indeed, you won the Colworth Medal in 1982. So okay. Yes. Sarah, on the other hand, has won the Colworth Medal in 2011. So Sarah, would you like to introduce yourself, please?
Thank you, Luminita. Yeah, that's right, 2011. At that time, I was a group leader at the MRC Laboratory of Molecular Biology in the Structural Studies Division. And that's because my sort of the area of research that I come out of is theoretical and computational biology, also called bioinformatics, using data science techniques and AI/ML, to study patterns in biology. And I sort of started off my independent career focusing on how protein complexes assemble, protein families, and combine then the computational methods with biophysics and structural biology methods to develop an interdisciplinary group to study pathways of protein assembly, and also use network theory as applied to gene regulatory networks. And that really then took me into regulation of gene expression and using T cells as a model. When next generation sequencing technologies came out, we started studying T cells with transcriptomics, epigenomics technologies, and combined with computation. And I moved to where I am today. I'm sitting here on the Wellcome Genome Campus, which is sort of just south of the city of Cambridge in England, at the Wellcome Sanger Institute. And I spend 1 day a week in the physics department in the Cavendish Laboratory of the University of Cambridge in theory of condensed matter, where there are theoretical and statistical techniques used in condensed matter physics that synergize well with the methods that we use in biology for sort of modeling systems and so on. So I head a department called Cellular Genetics, and I started that department at the same time as co-founding an international consortium called the Human Cell Atlas Project to map the cells in the human body using single-cell genomics and spatial genomics technologies coupled with data science. And that project now has over 3,000 members across the world, and I've mapped sort of 120 million cells in dozens of tissues in the human body, and happy to talk more about that later.
I'm sure we'll have opportunities for that, and that really sounds very impressive. Finally, Akhilesh, you won this award in 2012. So would you like to introduce yourself, please?
Yes, thanks very much, Luminita. Yeah, I mean, I think my career path is slightly different. So I started off at medical school in Cambridge, and really my entry point into science was a third-year project, and I was working on appetite control and the brain. So I was really interested in neurosciences, and that's why I eventually became a neurologist. But my interest in circadian rhythms came about peripherally. I was going to do a PhD on appetite control, but that got derailed slightly because my supervisor moved to Nottingham. So I kind of fell into the lab next door, which was working on circadian rhythms, which I was fascinated by, but never really got into until that point. And that's how I came to be in the field of circadian rhythms, which I've stayed in. I've moved slightly into a different field, which is sleep, since then, but that's how I ended up bizarrely in circadian rhythms, which doesn't necessarily fit with standard kind of ways of thinking in the world of medicine. And then I pretty much stayed in Cambridge for 20 years, then moved to the Crick for about 3 years, and then moved to Penn about 5 years ago. And really that was a move because of the presence of a large number of circadian and sleep researchers here. So it's like a real epicenter for that field in the world. And that's where I am today.
Thank you. Just listening to all the areas that you describe, it just shows that the real breadth of what molecular bioscience means and has sort of encountered in the last few decades. And I'd like to take you a bit, take a step back to where you are today and ask you to think and share with us one or a couple of important milestones that really you feel really shaped your either research interest or indeed career pathway in the way that it has led to where you are today. Now we're gonna do this in the order I think that you're on my screen. David, you're top left here, so would you share that with us? One important milestone.
I'm actually struggling to think of. I mean, I honestly, I think I've tended to stumble from one area into the next. I mean, I guess some people really plan out what they're going to do, and I never have. I mean, I've just followed my scientific nose for many years, and yes, I mean, you occasionally get something. One of the really important things is when you get something that really comes up, and you think, what on earth's going on there? It's very tempting to just throw it away, but some of the most interesting things can lurk in places like that. So never dismiss something that seems. And another thing, I guess, well, this is a bit of a parable. So I worked on something. It was a gene, a promoter in bacteria actually called Lu500, which seemed to be responsive to whether there were mutations in topoisomerase in the same cell. And at the time we were originally discovering this, we just really couldn't nail it down. And it would have been very easy to fudge it and sort of say, well, it sort of agrees with the way we think things are going. But actually, as it turned out, we were honest enough to say we really didn't understand it at that time. And then, you know, theories develop and so on, and new ideas came into the field. And about five or 6 years later, suddenly everything dropped into place. And the parable to that was, I'm so glad we didn't fudge it, because later we gave a complete explanation in the light of new ways of thinking, and you know, we completely honest about that, and I think that was a very valuable lesson in how to do science for me.
Thank you. And Ron, in your introduction, you already started talking about different important moments, you know, that shaped your career. Is there anything else that, you know, can think of now?
I entirely agree with David that the unexpected results are often the most informative. In other words, if you ask, is it A or B, and it answers A or B, you take a small step forward. But if you ask, is it A or B, and the answer comes back, no, it's Q, you take a much, much bigger step forward if you can find out what that means. So I completely agree with him on that. I probably had two embarrassingly fortunate bits of good luck. One of which was the first protein we discovered turned out to be heat stable. So you could almost purify it just by boiling an extract and finding it was the one that was left. And that turned out to be incredibly useful for working out how proteins get into the nucleus, because it even stayed pentameric on an SDS gel, which is almost unheard of. And a heat-stable protein which also resists dissociation with SDS is wonderfully useful to study, and it led us directly to discovering there was selective entry into the cell nucleus, quite unexpectedly. The second lucky break was in the same area of science, namely finding what was interacting with nuclear localization signals. And my postdoc, Dirk Gerlich, found an extraordinary control experiment he did produced a totally unexpected answer. And the unexpected answer was because the protein which recognizes nuclear localization signals, which we called importin alpha, turns out to have an endogenous His tag. So it binds to nickel columns selectively. So the first two proteins my lab discovered, one of them you boiled eggs and took what was left in solution. And the second one, you ran a nickel column and took the one protein that bound spontaneously without having to add a His tag. So some of these things, totally unexpected twists, can be extraordinarily useful and extraordinarily important in influencing what you do next.
It's really interesting that you are actually describing this as unexpected ones. It makes me think of what Louis Pasteur said, that you know, chance favors the prepared mind. So I think it's more than just unexpected occurrence to that. Sarah, do you want, you know, to sort of recall any of these important milestones in your career, or in your deciding the research area you're going to go to?
I guess one, you know, for computational biologists, and it's more about finding, you know, extracting nuggets from the data and finding meaningful, you know, distinguishing what's biologically meaningful and important from, let's say, the noise in large data sets. And that's part of that is, you know, using the right techniques and, you know, using sensitive methods. And there are, of course, you know, new techniques that come out in, you know, in the very early days of the PhD. When I was starting my PhD in the mid-90s, it was things like hidden Markov models. So those were machine learning models that were developed for voice recognition, for instance, and they were then taken into biology for distant homologies between sequences. And so we were able to use that machine learning technique to identify patterns of protein domains in the first completely sequenced genomes, and to identify, how in bacteria like E. coli, or in simple genomes like yeast in eukaryotic genomes, what are the patterns of domain combinations in proteins? How are they related to each other within genomes, across genomes, and so on? So it was really bringing data science, techniques from machine learning and data science into biology, and that was incredibly powerful and sort of opened up a kind of view of biology that wasn't possible before. And I guess now, the generative AI, deep learning and generative AI, and this kind of chat GPT is sort of revolutionizing our view of biology in terms of how cells relate to each other, how cells develop, how they respond to perturbations, using this very high resolution, very, very rich, large data from single cell genomics from the human cell atlas to understand development and identity of cells and how they sit next to each other in tissues to form functional tissue units in tissue microenvironments. So, it's really been kind of the development of data science has had a massive impact on biology in many, many different ways in my career, and the discoveries that they've enabled have been incredibly exciting. And so in cell identity, I guess the most exciting kind of moments have been in understanding T cell development and identity, in innate T cells, for instance, that are in human. We find them in embryonic and fetal tissues because the thymus has sort of different expression of enzymes, and then seeing those cells in artificial thymic organoids. So they're called innate-like T cells and have very particular T cell receptors, and understanding, where they are and how they migrate around the body and how they develop by an unusual mechanism of TT cross-presentation, was kind of a light bulb moment. And that was really driven by a young clinician scientist called Chen-Chu Suo in my group, who worked with me and Manny Clatworthy. More recently, there's been a kind of incredible moment of seeing cardiac pacemaker cells in the data. There are 50 tiny cells that were standing out from working cardiomyocytes, and they were just, we discovered them from 2 deceased transplant donor hearts that, again, a young clinician scientist working with a Japanese, James Cranley, working with a Japanese postdoc, Kazumasa Kanemaru, sort of pulled them out from a large data set just based on a few marker genes, and that allowed us to understand then new patterns of glutamatergic signaling of the pacemakers to the autonomic nervous system. And these cells are so important because they determine the beating rate of our heart. So they develop during pregnancy and the embryonic stage of the early heart development, and then keep on beating and firing spontaneously until the last sort of the end of life, when our heart stops beating. And they have this very unusual characteristic of this spontaneous firing. So anyway, so that was, in kind of came out recently in Nature, and was kind of incredibly exciting to sort of see that. So I guess it's yeah, and then sort of protein complexes was graph theory, which kind of came again from I would say more maths and mathematical technique development to study protein complexes and their patterns of how they assemble in vitro, but also in vivo in the cell. So I guess I come from that slightly unusual perspective, if you like, in biochemistry. Although my training is as a bona fide biochemist, so my degree is in natural sciences from Cambridge, and my part 2, so-called, like bachelor's, is in biochemistry.
Thank you. Ak, you obviously brought your clinical training into research, so once again, a different type of perspective. Any important milestone?
Yeah, I mean, I think it was probably important in terms of just quickly recognizing the ideal system in which to test the thing that we wanted to test pretty rapidly. Just because you kind of have experience of how the body works. You have to learn a lot about the body, obviously, to become a doctor. So you're just more familiar with all the different body systems and what examples there are of different things. Immune privilege zones, et cetera, in the body. So that stuff's just at your fingertips, if you like. And this came to the fore really when we were trying to answer this question about circadian rhythms. So when I first started out, there was a little bit known about mammalian circadian rhythms in terms of the clock genes. These are genes that oscillate with a 24-hour period. And really my interest at that time was in like global techniques like transcriptomics and proteomics to try and figure out all of the things that oscillate in human tissues or mouse tissues at that point and cells. But one thing that struck me, even when I was doing my PhD, I came across this interesting organism, which was the cyanobacterium. And it was a bacterium that had circadian rhythms, 24-hour rhythms. And that was really controversial at the time, because it's like, why does this thing that divides more rapidly than 24 hours need to have a circadian clock? And, it's dismissed actually by the field for a number of years, but eventually they found the molecular mechanisms, etc. And everything at that point was converging on transcriptional rhythms. So you had these gene circuits where so-called clock genes would get transcribed, they would oscillate, and then they would repress their own expression, and then they would gradually get degraded, and then you would release that repression, and then a whole cycle would start again, and that thing took 24 hours. So this was the predominant model. And the cyanobacterial clock at that point didn't really fit the mold. The initial model did, but then a lot of stuff emerged in the mid-2000s that proved, in fact, that the cyanobacterial clock, this bacterial clock, could actually oscillate in the absence of any transcription translation. In fact, you could just put 3 proteins in a test tube with some magnesium and some ATP, and you could get oscillations of that protein complex. So it's based on a kinase phosphatase cycle. So you don't need a cell, you just need a group of proteins, and you can basically create a 24-hour rhythm. So this got us thinking, it's like, well, could this happen in mammalian systems? This is a bacterium, obviously a highly specialized organism, but could you see the same phenomenon in mammalian systems? And, thought about different ways to, test that. And the ideal system that I came up with at that time was to look at red blood cells from human patients or people, volunteers, because human red blood cells don't have a nucleus, they don't have any organelles, they can't perform transcriptional translation. So naturally occurring cells in someone's body have this, unique property that you could actually try and measure circadian rhythms in. And that put us down this line. Obviously, we had to find out how to measure circadian rhythms in red blood cells and things like that. But we cracked all of that, and really that was a major move forward for us, and obviously the field as well, in terms of characterizing that you could get non-transcriptional rhythms in higher organisms like us. And really the insight that we got through that work was the proteins that were involved in these oscillations, which are a family of proteins called the peroxyredoxins, are extremely conserved. In fact, every organism on the planet, apart from some very unique type of bacteria like Borrelia, which is kind of on the borderline between a bacteria and a virus, it doesn't really kind of fulfill all the criteria of either. We found that basically this protein existed in a similar form in basically everything on Earth. So that led us to think, well, if this thing seems to be involved in human oscillations, maybe it's involved in oscillations in everything. And that led us to think about whether this same kind of mechanism involving redox oscillations could be present in essentially anything. And that's in fact what we ended up finding, that you could look at basically a tree, a bacterium, an archaeon, all of different kingdoms, and find essentially the same type of oscillations, even though they didn't share these transcriptional feedback loops that you find as the kind of predominant model that people put forward for how things work. So that was really the key insight, was thinking of, could this protein be involved in other organisms as well? And I think that's what we're most known for.
Clearly, these are fantastic ideas, and in a way, different ways of coming to them, very innovative ways that shaped, indeed, your idea, your careers, and shaped the thinking in that particular area for others. I'd like to move you a bit from the direct research interest into more the other ways in which your career has benefited, for example, from mentors, people not necessarily from academia, possibly outside. What sort of key impact or key person would you be able to briefly recall in terms of having an influence into your career? Ron?
I think I'd single out two who I had enormous benefit from at a very early stage. Namely, my first introduction to biology was just after taking what would now be GCSEs. And in the sixth form at school, I suddenly found this subject called biology. But we had two young, very innovative teachers. And they decided to ban textbooks. So there were no textbooks available to us. Instead, they used the money to buy a library of recent books. And the first one I took out was called Inside the Living Cell, by one of the people who was involved in the definition of individual histones and naming of the histones. And it was one of those books which, by the end of reading it, which took a very short time because it was totally gripping, I decided, okay, this is what I want to do. And it was really because of the imagination of those two teachers to expose us to real scientific books rather than textbooks, that I've felt immensely grateful to them thereafter. Beyond that, working with John Gurdon on nuclear transplantation was very influential on my career, as was someone who I think is one of the unsung heroes of British science, namely Lionel Crawford, who had a laboratory at what was then Imperial Cancer Research Fund and is now part of Cancer Research UK. And his laboratory was very loosely run in that we were encouraged to do whatever we thought would be most productive in a way that was very enabling and encouraging. And I once did a count of the number of people who finished up as Fellows of the Royal Society having come through his lab, and it's an extraordinary list. I'm not going to reel it off now, but so he had an immense impact on a whole load of other people coming through his lab, and I shall remain eternally grateful to him for that. And then one other name I have to mention was Aaron Klug. And during my time at the LMB, I wasn't in Aaron's division, but he took an enormously benevolent interest in this young guy trying to do strange things. And it was immensely encouraging to have come across him, and very, very supportive.
Okay. David, any particular figure comes to mind in terms of mentor?
Not quite in the same way. I mean, I never really worked with somebody, you know, an immense figure, let's say, who stimulated me in that way. But much earlier, at one remove. Let me tell you, when I actually going way back, I mean, I can remember my first chemistry lesson at school when I was about 10 or something. I think, you know, that's what I want to do with my life. And to me, molecules and chemistry was all I was interested in. Biology was not so interesting. But what made the huge difference to me was when the Nobel Prize in DNA structure was announced, and suddenly I could see, you know, as a schoolboy, I could see that actually biology was a molecular science. That completely transformed it to me. And when I learned that things like viruses could be crystallized, I mean, that was transformative. But you know, at one remove, what made a huge difference to me, back in the I guess it was the early 60s, Francis Crick wrote a series of just wonderful articles in Scientific American that were, again, accessible to a schoolboy. And, you know, he was talking about the coding problem, the genetic code, things like Benz's work on the R2 locus and all this sort of thing. And I just devoured these things. And so, you know, although, of course, at that time I didn't know Francis Crick, I got to know him later. That had an immense impact into my thinking at the time. And even though I decided I would read chemistry as an undergraduate to get a solid grounding, I did ultimately, clearly, go into biology. And, you know, a few people as a young faculty who were very influential and stimulating to me. One was the same person that Ron mentioned, Aaron Klug, was very supportive, and another in the US was Don Crothers, that came from this fantastic American tradition of physical chemists working in biology. But more recently, since I'm getting on now, shall we say, the people that have been immensely important to me are long-term postdocs. I mean, I've had people in my lab who have been with me for over 20 years, and, you know, nominally postdocs, but really they're colleagues and friends, and they're tremendously important. I mean, there is, for example, Tim Wilson in my lab, who is a postdoc, but, you know, he's been with me for probably 25 years, I think. Every day in the morning, we go and have coffee together, and you know, 9 mornings out of 10, we're probably talking about ribozyme mechanism. You know, it's like that constant stimulation is important. And the other people are tremendously important to me, more and more actually, are my collaborators, and particularly collaborators in China, for example, Huang Lin, who is a faculty member at Zhongshan Dashi in Guangzhou. You know, we Zoom on a sort of a weekly basis or more, and I probably go to his lab. I'm going there in a few weeks' time again. Obviously, that was disrupted during the pandemic, but now we're back to 4 or 5 times a year. So these are the people that are important to me now.
Thank you. Sarah, anybody or a network that you think was particularly important for you?
I guess, you know, I obviously owe a lot to my chemistry teacher at school, Walter Henderson. I went to school in Germany, to a European school, sort of state funded. And he had a club basically of chemistry and biochemistry projects that he offered students. And that really got me very excited about this sort of secret invisible world of molecules that are, you know, these invisible machines, you know, enzymes and cells and so on that are the fundamental units of life, if you like. I guess later, you know, when I started my group, my first PhD student is Madan Babu, and at LMB, then there's sort of a few years later, after I had tenure, actually during the time that I was awarded the Colworth Medal, we had a kind of trio of faculty members in the Structural Studies Division at LMB. That was Cyrus Chotia, who was my PhD mentor, actually incredibly influential on me, myself and Madan, and, you know, similarly, we would hang out together, go for lunch, talk about, you know, the research that we were doing. And I agree that it's, you know, it's the intellectual exchange, but there's also a camaraderie, there's a friendship, you know, it's important kind of in terms of your well-being and so on to have these networks, and it's really lovely. And later, of course, you know, my postdoc mentor was the legendary Janet Thornton, when I was a postdoc at UCL, and she then was the director of the European Bioinformatics Institute and recruited me back as a faculty member, and I was the first joint faculty member between the European Bioinformatics Institute and the Wellcome Sanger Institute. And the network there around single cell genomics technologies kind of, this is after I was awarded the Colworth Medal, kind of 2012, 2013, you know, I moved over here to the Wellcome Genome Campus, And we had a sort of triad of John Marioni, Ollie Steger, and myself that was kind of similar. So we all had offices next to each other. And that was incredibly exciting because it was the era of explosion of these technologies and development of the computational methods that were allowing us to sort of see cells, with their complete molecular fingerprint and their complete molecular identity, in a systematic way for the first time. And that era was also, Ollie and John and me being sort of next door to each other and having the exchanges between our groups was amazing because I had the genomics, John had the statistics, Ollie had latent variable models, machine learning, and that combination was incredible. I would say the one person that who really did change my life was Carol Robinson. As an external collaborator, like outside of my institution, and that was because she invented macromolecular mass spectrometry, and her method allowed us to probe protein complexes and their associations, their non-covalent associations, using this quantitative technique, native mass spectrometry, that Carol developed. And so we were able to show that the order of protein assembly, not only in evolution in terms of conservation of subunits being associated, but also assembling into larger soluble globular complexes, follows a stereotypical pathway that's determined by the buried interface size. And it's sort of the same principle as protein folding, where you have hydrophobic residues buried in the protein core. And Carol's technique allowed us to show that interface size determines assembly in vitro, because she was able to take complexes and disassemble and reassemble and probe their mass. And so she showed experimentally what we predicted computationally, if you like. And that proof, that kind of experimental proof, then gave the whole theory support. And we had a whole body of work, many publications in Nature, Science, Cell, and so on, showing this together. And Carol's just an incredibly inspiring kind of role model. I'm not sure if she was in fact, possibly a nominator, or maybe it was Janet Thornton for the Colworth Medal, and has basically also always had that, ambition of also, getting recognition and awards, and continuing to kind of have the energy to sort of push for these things. And I really owe this to her, and to a huge extent, that to kind of keep going in that front. And it is worth mentioning in this context, just sort of, that Sheena Radford was the only woman who was awarded this medal before me, and that was in the mid-90s, I think. And then sort of 50 years later, I came along, so 10 or so years after Sheena. And it's really the people who nominated me that I owe that to, and obviously 50 years of the Colworth Medal, 2 women, you can see you don't have to be a theoretical physicist or a statistician to see that there's a statistical imbalance there. And so I really owe my nominators, and I'm really glad to see that there are 4 women who've been awarded recognition in the last 10 years, and the society and the community is really moving forwards, and that's incredibly gratifying.
That indeed touches to something that hopefully we'll have time towards the end to put in terms of nominations and so on. Ak, I'm aware that you haven't had the chance to actually say something about your mentors, just very briefly. There is one other main aspect that I want to push you after that, so I'm looking.
Okay, cool. I'll keep it brief. Yeah, I've had a variety of mentors at different stages. I remember undergraduate, I was interested in neuroscience, so Barry Everett at Downing College in Cambridge, where I was doing my undergrad, was hugely influential then in kind of getting me to think about science more broadly. And he was a big influence, I think, throughout those years. I think later on I needed different guidance. So Mick Hastings was my PhD supervisor, and he was like really obviously formative in terms of how I think about science. But his mode of science is a bit different to mine as well. So I've kind of diverged a bit from him in terms of how I think about things. But I think he's obviously still hugely influential. He's a great friend now as well. And I think, again, speaking to what Sarah said, you have these enduring relationships and camaraderie, and that sees you through life, I think, because you encounter obstacles all the time. And I think after that, I've had a mixture of different influences and mentors. I think one of, obviously, in terms of navigating clinical medicine and research, Steve O'Reilly was hugely influential. He started this new institute, and I was lucky enough to get my first lab there, essentially, with the help of a Wellcome fellowship. So he's been hugely influential. I think he's one of the ones that nominated me for the Colworth Medal. And I think around that time as well, because of my, I had a fellowship at St. John's College, and at that time, Chris Dobson, the late great Chris Dobson, was the master there, and he was hugely influential in terms of just general advice, the sagely advice about the world. In fact, he told me stories about Carol Robinson and all of the issues that she faced, because he was at Oxford when she was at Oxford as well. And it's she's an inspiring character from multiple standpoints. I don't know her, but I know vicariously of her through Chris talking to me about her, and his general advice was amazing. And I think more recently, Keith Peters has been a huge mentor for me as well. He's a big figure in UK bioscience, and he's been really helpful in terms of advising me on different things. So yeah.
Thank you, Ak. We've spoke about ideas, brilliant ideas, how they can come, expected, unexpected. We spoke about people. We spoke about your achievements and other people's achievements. We quite often encounter difficult moments in science, and people will say that science is a very difficult subject, albeit so enjoyable for many of us. Now, there must have been some obstacles or challenges along your way. And again, just towards us needing to wrap up in the next minute or so, I will want to ask you to reflect a bit and to share on where the obstacles may have come in your careers. What do you see the role, and whether those challenges or obstacles, do you see them different in today's day as opposed to what has happened decades ago? Ask Ron to start again.
Identifying obstacles, I tend not to keep them in my memory, but I suppose one self-inflicted obstacle very early on, when I was still an undergraduate, is I started writing and performing songs, and doing this in 1960s folk clubs got really quite exciting and interesting. And as a second-year undergraduate, I got really quite carried away with the apparent success of this activity, and was asked to do a double bill, which was rather formative, because it was with someone I'd never heard of, and I was on first and thought it had gone really well and was really feeling very smug about how things had gone. But he then, as I sat down, and he followed, and he started with words that I could still recite in shock, with a song which was utterly extraordinary for its time. And he was Paul Simon, of Simon and Garfunkel fame. When Art Garfunkel couldn't get a work permit, he was working on his own. And I was completely carried away with this and yelling and cheering like all the rest of the audience, until I remembered it was a two-part concert, and that I had to follow him when he stopped, because I had to do my second half and then he'd do his second half. And following Paul Simon was quite a challenge to a 20-year-old undergraduate. So that evening, I very firmly decided it was time to start studying a bit better, and realized that my future career was going to have to lie in science, because it certainly wasn't going to succeed in music. So it was an unconventional obstacle, but a very, very productive one.
So it's as important as everything else to know where not to go.
Exactly.
Yes, David, just any challenges, especially if you see challenges today have changed, but what are challenges that science in the UK face?
before I answer that, Ron and I ran a conference, Harden conference together many years ago. And at the conference dinner, Ron had his guitar and played these songs. So I remember them very well. I think he could have had a career in that, but hey. No, I mean, for me, obstacle, that's absolutely clear, the pandemic. Unfortunately, you know, I was funded by Cancer Research UK for 30 years continuously, and our renewal year was 2020, and CR UK lost hundreds of millions in the pandemic. It wiped my lab out. All my Chinese postdocs went back to China, and my lab's really shrunk now. So I'm just building up again. But that's been a huge obstacle, which I'm still fighting my way back from.
Thank you. Sarah, about yourself?
I mean, I guess, you know, there have been like obstacles starting from, you know, when I came to Cambridge as an undergraduate from a different school system, you know, as a foreigner from a different school system without the same specialized A-level preparation to, you know, starting my group in LMB as a computational biologist in a structural biology department that was geared more towards X-ray crystallography and so on. So, you know, to the pandemic was obviously a huge obstacle, but at every point, the question that you need to ask yourself, I think, when you're faced with an obstacle is how can you turn lemons into lemonade? How can you make the best of, you know, the situation that you're in? And how can you turn that to your advantage? And, you know, a big challenge was leaving LMB where I had tenure, you know, I'd been for a dozen years and coming to the Genome campus to this new environment. I went from this very Nobel-laden, very prestigious place and decided to leave, you know, which was, a lot of people said it's a mistake. No, I thought about it for a long time. And then here, you know, then new opportunities opened up that I never dreamed about, like Human Cell Atlas and you know, the voyage through the human body at the cellular level. So I think, you know, it's what you said, sort of seeing the opportunities, you know, what you said before, Luminita, I think that's what life is about.
And I'm hearing sort of to not be afraid of taking bold steps.
Exactly. Yeah, sort of jumping into the deep end of the pool, the swimming pool and swimming, yeah.
Ak, anything, you know, that you would like to share?
Obviously, tons of obstacles in the way. I think, you know, people, you know, often looking from the outside think that, you know, it's like a duck on water, you know, they're scampering underneath to move, but you just see the gracefulness on the surface. And that's very much the same with scientists. You see the CV and the achievements and stuff, but you don't see the hard work that's gone on under the hood to do that and all of the obstacles you faced. And I think, you know, there's tons of obstacles I could mention, you know, funding, for example. You know, when I first got my Wellcome fellowship, I was starting off and I thought, oh, I've got a grant, excellent. Maybe I should write some more. So I wrote like 6 over the next 2 years. Every single one of them failed. But, you know, that was a formative experience because it taught me how to write better grants, but also that some of it is a bit stochastic, quite frankly. You know, the system is not the best. No system's the best, but you have to just try and keep going and, you know, have this perseverance. And I think that's one of the things that comes out in most scientists that you speak to is perseverance and tenacity, you know, just persisting when you are up against obstacles, no matter what they are. And another thing that's stuck in my mind from conversations with Steve O'Reilly were that sometimes, you know, when you're getting stuck with institutional things, which happens a lot, you know, there's always demands on space and everything else in institutions, he said sometimes you just have to mold the situation to fit with what you need rather than trying to change it completely. And I think, you know, that's something that I often come back to when I get frustrated with things. So there are 2 bits of things that stick in my head in terms of obstacles and, you know, tactics or strategies to overcome them.
Yeah, what you're all saying almost make me think that we will need another webinar simply to look at this. We'll achieve a lot of good advice for anybody trying to build a successful career in biosciences. But to wrap up our discussion, you know, I will go to one of the questions that we received, which links very well with what Ak was just saying, and that is, what advice would you give to biochemistry students starting on their journey, especially to assure them of the future? Very briefly, will have to be Sarah.
Sure. you know, look for be imaginative and look for the opportunities in interdisciplinary spaces, because that's where a lot of creativity lies. And also, you know, find great collaborators and work together as a team, because teamwork makes the dream work. And nowadays we're seeing more and more of that, and that sort of fits with this concept of requiring different disciplines to work together. And that's, I think, the future of biochemistry.
David, your advice to biochemistry students, or indeed early career researchers in biochemistry?
I totally agree with what Sarah just said. I think I've always worked at the interfaces between areas. I mean, originally in my PhD between mathematics and physics and chemistry, but I've always found that's where the interest, it's not true for everybody, but for me personally, I've always thought that was where the interest was. The other thing I would say is, look, follow your passion. You know, we're paid to do this. We're paid to just follow our noses in science. Don't worry about what awards you're going to get, whether you're going to get a paper in a prestigious journal. Follow what's interesting. Follow your nose in science, and you can't fail to go wrong if you do that.
Ak, what would be your advice?
Yeah, I mean, I would definitely echo both what Sarah and David said about collaborations, but also passion for something. I think, you know, fundamentally you have to be interested in what you're doing to keep you going through obstacles that come in your way all the time, and failures. You know, most of the stuff that we're doing, at least in wet bench work, fails badly often, and so, you know, you're going to hit that every day. So you need to be interested in what you're doing and have a passion to keep it going. And I think, you know, when you're choosing what area to go in, you know, don't get obsessed with it, because, you know, you can always change tack. You can move from things, and often that's the way things work out. You know, you start off somewhere and you end up somewhere completely different. And I think you just have to sometimes ride the wave with things and not be too strategic at an early stage, because you might close off doors, which could lead to opportunity later at an early stage.
And let's end with Ron's advice for budding biochemists.
First, I would firmly endorse what my three colleagues here have said. I think the importance of collaboration, the importance of interdisciplinarity, and the importance of looking for the unexpected result and trying to understand it are all vitally important. But I would add one further thing, which is, for many years above my desk, I had a quotation from the French philosopher André Gide, which read, "One does not discover new lands without agreeing to lose sight of the shore for a very long time." And I think that that's also a very important aspect. It's become increasingly difficult to maintain that principle with current funding pressures, but it is an important one to remember that you don't make big discoveries by sailing along the coast. You have to have the courage to sail out into new territories if you're really going to reap the full benefits.
Thank you very much for that, and thank you all for sharing all these insightful ideas with us. I will remind everybody that you can continue the conversation online by following Biochemistry Society and Portland Press Publishing on Twitter. In direct relations with today's focus, I would like to remind everyone that the Biochemical Society 2025 awards are now open for nominations, with the deadline being 1st of November 2023. And here would have been the advice that Sarah has given. Hopefully that will increase the diversity of those nominations. Please do look out on our website for all future upcoming webinars. Join us on the 19th of October at 3 o'clock British Standard Times to watch the webinar entitled Alternative Protein Careers for Biochemists. And finally, I like to highlight that it's more important than ever to stay connected and engage with your fellow biochemists and bioscientists. It is indeed an exciting time to join the Biochemical Society, the whole community of researchers and specialists, and to stay connected and take advantage of key benefits. It is again my pleasure to thank the panelists for sharing their insightful stories with us. Thank you all for attendance, and hopefully see you at some next events that the Biochemical Society organizes. Thank you. Bye.
Thank you.
Bye. Thanks.